Have you ever stopped to think about your body as this incredibly complex machine running on, well, a kind of digital code? In a lot of ways, it is. Today, we're going to dive into what happens when there's just a tiny, single-letter typo in that code, a mistake that can have absolutely massive consequences for your health. Let's get into it.
Okay, so you probably remember from high school biology that mitochondria are the powerhouses of the cell, right? They're the little engines that generate almost all the energy that keeps us going. But here's the really wild part. These little engines have their very own instruction manual, their own unique DNA, completely separate from the main genetic blueprint in our cells. So, that begs the question, doesn't it? What happens when that special instruction manual has a typo? Well, as it turns out, the consequences are way, way bigger than you might imagine.
Now, these little genetic typos, they have a proper scientific name: single-nucleotide variants. And our whole story really starts by understanding just how tiny and yet how incredibly powerful these little errors can be.
A single nucleotide variant, or an SNV for short, is exactly what it sounds like. It's a single-letter mistake in the genetic code. I want you to picture an instruction manual that's, say, 16,569 letters long, and SNV is just one of those letters being wrong. Seems pretty insignificant, doesn't it?
Yeah, you'd think so, but you'd be wrong. It turns out that even one single incorrect letter out of nearly 17,000 can cause that entire cellular engine to just sputter and fail. These tiny typos have been linked to a huge range of serious health issues, everything from diabetes to heart disease to some really serious neurodegenerative disorders. It's pretty clear that even the smallest mistakes can have the biggest, most devastating consequences.
So how in the world do scientists actually hunt down these specific typos and figure out what kind of damage they're causing? Well, the process is this fascinating piece of digital detective work. So the first step was a massive hunt for clues. Researchers basically cast this huge net. They started with over 1,700 scientific articles. Then they had to sift through all of them, narrowing it down to the 167 most important studies. And from those, they painstakingly extracted every single reported genetic typo that was linked to mitochondrial problems.
And after all that digging, they had a list of suspects. 56, that's the number. 56 specific SNVs, or typos, that kept showing up again and again in studies about mitochondrial issues. You could call it their most wanted list. Now, with this list of 56 typos in hand, the investigation goes digital. And this is where it gets really cool. Instead of spending years and years in a lab testing each one, the researchers used powerful computer software to run, essentially, a digital crash test simulation for every single typo.
They used this whole suite of really sophisticated digital tools. You had AlphaFold, which could predict the 3D shape of proteins, the actual machine parts of the cell. Then there's RNAFold, which predicts the structure of something called tRNA. Think of those as the assembly line workers that help build the parts. And finally, SwissModel, which could analyze how a typo changes a protein's basic chemical properties.
And the results of this whole digital investigation, honestly, they were stunning. You could literally see how a single typo completely warps the essential machinery that keeps our cells running.
Okay, check this out. This slide shows what happens to a really key protein called mTND1. On the left, you see the normal, functional shape. That's the blueprint. But look at the right. That's the result of just one typo. The protein is all misfolded, it's warped, it's like a perfectly good machine part that's been bent out of shape, and now it just won't fit where it's supposed to go.
And here, you can see why that protein gets so warped. The typo actually changes its fundamental chemistry. So, for example, that T3394C mutation makes a part of the protein that's supposed to repel water suddenly attract it. And that single little change in its relationship with water forces the entire protein to fold completely differently, messing up how it connects with everything else.
Sometimes the damage is even more dramatic. I mean, look at this MTCYB protein. One particular mutation doesn't just warp the part. It sticks a premature stop sign right in the middle of the instructions. So instead of a full, complex protein, the cell just makes this useless shortened stub. The machine part isn't just bent, it's been chopped in half.
And it's not just the machine parts that get messed up, it's the assembly line workers too. This is tRNA. Its job is to carry the building blocks to make the proteins. On the left, you see its normal, precise cloverleaf shape, but on the right, you can see how mutations just totally warp that structure, especially at the acceptor arm, the exact spot where it needs to hold on to its cargo.
But it's not just about a wonky shape. As this chart clearly shows, these mutations make the tRNA molecules dangerously unstable. The normal version, the wild type, is very stable. But add one mutation, and especially with two, the stability just plummets. This means these crucial assembly line workers literally fall apart much faster and just can't do their jobs.
So yeah, understanding these specific typos is fascinating stuff, but this research gives us something even bigger, even more important, a completely new way of investigating genetic diseases.
And this right here, this is the real game changer. The true breakthrough isn't just the data about these 56 specific mutations. It's the creation of this repeatable digital method, this biochemical pipeline, that scientists can now use to quickly analyze any new mutation they happen to find.
I mean, the power of this pipeline is just immense. It lets researchers predict which typos are probably dangerous, which just dramatically speeds up all kinds of research. It can help them design way more accurate diagnostic tests, and maybe most importantly, it can guide the development of drugs that are targeted to fix the exact problem caused by one specific typo.
You know, the researchers themselves put it perfectly in their paper. They called this method a powerful tool in the arsenal of precision medicine. And that's exactly what it is. It's about moving away from one-size-fits-all treatments and getting closer to medicine that's tailored to our individual genetic code.
So what does all of this cutting-edge science really mean for you, for me, and for the future of our health?
Well, work like this is a huge step towards a future of truly personalized medicine. We're talking about a future where we can look at our own unique genetic blueprint, find the potential typos, and understand our personal health risks on a level that was, frankly, science fiction just a few years ago. Which leaves us with the final, kind of mind-blowing thought. As this technology keeps getting better, could we one day get a report on our own personal mitochondrial typos? And could we use that very information to predict and maybe even prevent health problems down the line? All the science seems to be pointing in that direction.